Hello everybody. Today we are going to start with the new chapter in biology, that is chapter number 5, Transpiration. So this is the second chapter from the section of botany in your syllabus. The first chapter being absorption, which we just completed, and now we start with the new chapter, that is transpiration. So transpiration is we can see that the plant, it requires water for its general purpose, general use for its producing for doing the photosynthesis, for also taking the minerals right from the ground to the upper part of the plants. So water is required for the plant, and accordingly what will happen is that the upper layers of the plant, the upper aerial parts of the plant, that is the leaves and the stem, they are going to lose certain amount of water, as we can say evaporation, or the we can say that loss of the water from the aerial parts of the plant is called as transpiration. So transpiration is the water which is going to be lost by the plant from the aerial parts, that is the shoot, that is the leaves and the stem. So when the plant is going to lose water from its cells, then it is going to be said to be as transpiration. So all plants continuously absorb water through their roots. This water is conducted upwards to the stem and is distributed to all the aerial parts, including the leaves. Only a small quantity of water, about 2%, is used by the plant in photosynthesis and other activities. The rest of it is almost lost to the atmosphere as water vapor as a result of transpiration. So it is seen that out of the amount of water which the plant is going to absorb from the soil, only 2% of the water is going to be utilized by the plant for photosynthesis, or else other complete 98% is going to be lost by the plant in terms of transpiration. So transpiration becomes a very important source by which the water is being lost by the plant. So transpiration is the loss of water in the form of water vapor from the leaves and other aerial parts of the plant. So that is the definition, or we can say defining transpiration as transpiration is the loss of water in the form of water vapor from the leaves and the aerial parts of the plant. So the upper layers of the plant, aerial part means everything which is above the soil. Above the soil will be the roots, and they will be the shoot, and that will contain the stem and the leaves. So, and of course, so stem and the leaves are the one which are going to give rise to transpiration. Now we have got a small experiment to demonstrate this particular thing. So in this experiment, is a very simple experiment. We have been doing it like from the 7th standard, I suppose, where you take a pot, and that pot is having a plant. So there is a well potted, it's a potted plant. So this plant is, let's say this is the plant what we are talking about. So this is a plant, okay. So what we do is that to this plant we are tying a plastic bag, a polythene bag is tied over here, such that you are covering only the aerial part. You are not covering the pot, neither the soil. The soil and part is going to be remaining like this, so that you are sure that whatever the water droplets what are seen over here will be due to the aerial parts and not due to the soil leaving out the water, or soil evaporation of the water which was present in the soil. So that is, we don't want that to happen. That's the reason we are tying it up only to the aerial part of the plant. We are tying a plastic bag to it, and then you are leaving this thing in the sunlight. So in the sunlight, you can see that after a few hours, there will be droplets. There will be small droplets of water in this particular area. So these droplets are the indication that there has been transpiration taking place, or we can say loss of water taking place by the aerial parts of the plant. So so that's the experiment. Take a medium-sized, well-watered potted plant. Cover the plant with a transparent polythene bag and tie its mouth around the base of the stem. Leave this plant in sunlight for an hour or two. Drop of water will soon appear on the inner side of the bag due to the saturation of water vapor given out by the leaves. The water vapor condenses only if the outside temperature is cool enough. Of course, if it is too hot, then it will not condense, it will remain in the form of the vapor only. But if it is outside temperature is cool enough, then you will see that this water vapor which is over here is going to get condensed, and there will be droplets of water on the bag. A similar empty polythene bag with its mouth tied and kept in the sunlight will show no droplets of water. So if we are not keeping the plant, okay, there is no plant, but we are just keeping the polythene bag over there, do you see any droplets of water? No, you don't see any droplets of water. Why? Because it is just to prove, it is an experimental setup. So experimental setup will be one in which you are going, and the control will be the polythene bag without the plant in it. So that will be the control setup. So this is the control to show that plants transpire water in the form of vapors. If tested with dry cobalt chloride paper, the drops will be confirmed as water only if the blue paper turns pink. So this is a test for the moisture. For test for moisture, if you want to find out that whether the vapors or whatever the droplets of watery feel is there or that liquid is there is water or is something else, we are going to take cobalt chloride paper. Now cobalt chloride paper is normally blue in color, and when it comes in contact with moisture, when it comes in contact with water, it turns pink. So we've seen that cobalt chloride paper it turns from blue to pink in presence of water. Okay, so the cobalt chloride paper turning pink from blue is the we can say the test to identify that the droplets of the liquid which is seen inside this is water. So that is how we are confirming this thing. You can have another experiment also. Arrange three setups A, B and C as follows. So they are given three things over here which is connected over here like this, 1, 2 and 3. These are the three connected things over here. So see that setup A, take a small well-watered potted plant, preferably one with broad leaves. Enclose the pot completely within the polythene bag and tie the mouth of the bag firmly around the base of the stem. This would prevent the escape of water vapor from the pot. Now cover the entire plant with a bell jar as in the figure 1. So you can see that first of all you are taking the complete pot, so your complete plant. So we are taken enclose the pot completely. You are completely enclosing the pot. Why? Because you don't want the pot ka pani to evaporate. You don't want the evaporation of the pot ka pani. That's why you are, instead of tying this part, we are tying this part. So you are sure that whatever water vapor will be there in the bell jar will be because of transpiration, and this one because it is already covered in the plastic bag. So whatever evaporation would be taking place over here will be within that plastic bag and not going outside. So that's the only thing, that we are taking a well potted plant and we are covering the pot part, the base part, as the root part along with the pot is going to be covered with the plastic bag. So you are covering it with a plastic bag firmly so that you are not you are preventing the escape of water vapor from the pot. Now you are putting this whole plant in a bell jar. So you're taking this pot and taking a bell jar and keeping it on top of it. So a complete bell jar is placed on top of it. So it's something like this. You are taking suppose this is a potted plant, and this potted plant is covered with, first of all, is a polythene bag, and then you are taking a bell jar. So the bell jar is taken like this. This is the bell jar. Okay. So the bell jar which you are taken over here. So this bell jar is kept on top of this particular pot. Okay. So that is one arrangement, that you are taking a bell jar and placing it over. You are going to have a setup B. Arrange the same another similar plant and cover it with bell jar exactly in the same manner as in the first, except that here you also keep a dry cobalt chloride paper by the side of the plant inside the bell jar. So the paper may be pinned to a wooden stick or a strip or a cork sheet, kuch bhi. You are basically trying to see to it that you are placing a cobalt chloride paper which is blue in color. So the blue color strip is there inside that particular bell jar. So यहाँ bell jar में you are placing this cobalt chloride paper. So you are going to keep a cobalt chloride paper over here. You can pin it or place it or put a sellotape and put it whatever. It's not a important thing to be seen. And setup C will be take a third bell jar without the plant but in still containing a similar piece of cobalt chloride paper. Now keep all the three jars together in the sun. So you are having one arrangement like this with the cobalt, one arrangement without the cobalt chloride paper, one with cobalt chloride paper, other one without the pot. Okay, so that will be a control setup. Control setup will be the one in which there is no pot but there is this cobalt chloride paper in it. After about half an hour, we observe the first bell jar should show water vapor condensing on its inner wall. So you will be finding the droplets of water on the inner walls of the bell jar in the first case. Okay. In the second bell jar would also show the similar condensation at the same time. The initial blue colored cobalt chloride paper will turn pink. So this one over here is going to turn pink. So this blue color one is going to turn pink in the second condition. And then the third condition, the cobalt chloride paper has remained the same blue and there is no change in the third one. So the blue color of the cobalt chloride paper in the third bell jar does not change at all and there are no water droplets on the jar inner walls either. So we can see that the three conditions were one whereas you see the condensation of the water droplets on the bell jar. Second one, yeah, there is a condensation of water droplets in bell jar plus the cobalt chloride paper which we had placed over there is also going to show that the condensation is of water because the cobalt chloride paper had turned from blue to pink. And the third one is the control setup wherein you know nothing is going to happen. There is no change taking place. The cobalt chloride paper is still blue and there is no condensation of water anywhere on the bell jar inner surface. So the third bell jar is the experiment. In the experiment is a control which proves that there is no moisture in the air due to transpiration as there was no plant in it. So there is no moisture in that particular air of the bell jar because there is no plants as though there is no transpiration. By taking the three bell jars as described above, there is a double visual proof of transpiration. First, condensation water vapor into droplets and change of color in the cobalt chloride paper. So you can see that here we are proving two things. One is that there is a condensation taking place of the droplets of water and that particular thing is water. We are proving it by the cobalt chloride paper. So the cobalt chloride paper is going to tell you that the condensation is of water and not some another material. So this way you are proving two things, one double visual proof. So it's a double visual proof. One is a visual proof that there are droplets of water and second visual proof is the presence of the cobalt chloride paper. So both the things are being proved in this particular method. So we see that the cobalt chloride paper which is dry, it is blue in color, but it is when it comes in contact with moisture, it will turn pink. So that is an important observation to be seen and which is going to be one of the important questions they would be asking you if they ask you this particular experiment as a question in the examination. So they may give you this experimental examination and they will tell you certain 5 questions based on this. So first thing would be what is to be proved, what is the aim of the experiment. So we are going to say that we are trying to identify the process of transpiration. Then they will say define the process. So we'll have to define transpiration. Third question would be what observation can be seen. Okay. Then fourth question, why we are not covering this thing. There could be many different questions which could be asked. Well, what will happen if this is not placed in the sunlight but it is placed inside the room? Then what would happen? What would the change? What would be identified? Why did it put in the sunlight? So what are the factors which are affecting this thing? So there could be many questions could be asked based on transpiration connected to this particular experiment. We now see that. So we understand one thing, that transpiration does take place. So that we have understood, the transpiration does take place. But how much amount of transpiration takes place, that is something which need to be measured, because many times we require that we should know that how much a plant is going to transpire, so that we can know that how much amount of water has to be given to that particular plant. So if the transpiration rate is more, we'll have to water the plant much regularly. If the transpiration rate is low, you may not water it that regularly. Anyway, this is just an example, but mainly we do study that what is the rate of transpiration, and to measure the rate of transpiration we use many methods. We are going to study here two different methods. One is the weight method and other is called using a device called photometer. So these are the two things what we are going to have. So let's start with the first one, and that is the weighing method. So there are two number of methods of measuring transpiration. Some of these are there. Sorry, there are number of methods, not two. There are number of methods to measuring transpiration. Some of these are, first one is weighing method. So in the weighing method, it's a thing that we are going to take a potted plant, okay, and or you can just a jar containing some water in it and the plant in it, and then you are going to weigh that thing, and then you are going to weigh it again after some time. You will find that the water level has been lost, and there is the transpiration has taken place, and therefore there will be loss of water. So let's understand it properly. Let's see this thing. A small lightweight potted plant can be weighed before and after the end of a certain period of time. Okay, so what we are going to do is, before and after a certain period of time, you are going to weigh the mass of a potted watered plant. The soil surface and the pot should be carefully covered to prevent evaporation from the surface other than the plant. The loss in the weight of the plant during that time is due to the loss of water by transpiration. So we can see that what we are trying to do is that we are trying to ascertain that the soil which is there in the potted plant, if at all that loses water, that particular water should not be evaporated. It should remain there. So that's why we are going to see to it that the lower part, that is the pot area, is tightly sealed, so that you are not having any evaporation of the water from those parts. So whatever is the loss in the weight should be proved that it is only due to the loss of water due to transpiration. So that is what we are trying to do. So we are trying to take a pot. We are going to see to it that that pot का जो नीचे का part है, it is well covered and tightly packed, so that the vapors of the water present in the soil doesn't evaporate out and it cannot move outside. So water is remaining inside only, and so transpiration is the only loss of water which could take place. And that transpiration loss, if it is measured accurately, we come to know that what is the amount of water which is lost due to transpiration by a plant. So that was the thing, and now they got a slight improvement in this case, and this is an improvement in the weighing method can be made by using a glass bottle with a graduated slide tube filled with water and a tube fitted to it as shown in the figure. The water level in the side tube falls to demonstrate loss of water through transpiration from the leaves. So that is what they have done is, we have got a potted plant, so there is a sealed bottle. So there is a sealed bottle, and the sealed bottle over here, they are having a plant which is put inside. So there is a roots over here and the plant is over here, and there is a small kind of a tubing here. So what is the water content over here? The same level is shown over here. So of course we see that initially the water level was up to this point, and then when the whole apparatus is kept in the sunlight for some few hours, you will find that there is a drop of water over here. We know that water cannot escape from this part because it is not allowing it to go out. So whatever is the loss in the drop in the level of the water over here, it shows that much is the amount of transpiration given out by the aerial part of the plant. So that is what is a modified version of this experiment. So the water level in the side tube falls to demonstrate loss of water through the transpiration from the leaves. So that is the thing. Another thing is, this would indicate the volume of water loss that can be compared with the loss of the weight with the help of weighing machine or by converting cc into grams. So we can see that by the volume of the water loss can then be converted into the weight, so loss in weight, and with the help of the weighing machine we can find out the difference between the cc and the grams. So that is what is the setup over there. Then there is another alternative method of weighing system where you are going to take a small twig. So here another weighing experiment can be made by using a test tube filled with water, inserting a leafy shoot, no roots in it, and pouring some oil on the surface to prevent loss of vapor by evaporation, loss of water by evaporation, and place the test tube in a small beaker and weigh them together. So we are taking a test tube. So it is a condition in this way that in this method you are taking a small test tube, a test tube is taken, and then water is there. All the water you are taking some oil layer, so this is the oil layers, this is oil and this is water, and you are going to place a stem, so which will be a twig. In fact, there are no roots, only the leaves are there. So there will be only the leaves over here, and you will be weighing this particular test tube. So you weigh the test tube completely along with the water and oil and the plant, everything. You weigh this particular thing and keep it aside for some hours, and then weigh it again. You will find that there is a loss of weight due to transpiration. So place the test tube in small beaker and weigh them together. Remove the intake test tube and keep it straight into the test tube stand for a few hours. Weigh it again by keeping it in the same beaker. Any difference in the weight is indicated a loss of water by, so any loss in weight, so whatever is the loss in weight, so suppose पहले वो 25 grams था, अभी वो 24 grams हो गया, तो 1 gram is the loss of water by evaporation. We'll indicate the loss of water by the shoot due to transpiration. Since there are no roots to activate absorb water, the water loss through transpiration is going to be much less. Because if there would be roots, yes, it would be much faster. Because we are not taken roots, therefore the thing is going to break will be much less. The amount of water which will be given out will be much less. Now we come to the most important part of the this one, and that is the photometer method. So next one is the measurement of the transpiration using a photometer. So photometer is a device which is used to measure the rate of transpiration. So the photometer means a device that measures the rate of water intake by a plant. Photo, that is drink, and meter means measure. So that is water intake is almost equal to the water lost through transpiration. So normally, yes, of course, it is not going to tell me exactly the rate of transpiration, but it is going to tell me the rate at which the water is taken in by the plant. But as we know that whatever water is taken in by the plant, उसमें से सिर्फ 2% जितना है जो photosynthesis वगैरह में use होता है. So it is only 2% which is lost in due to photosynthesis and used up by the plant for photosynthesis. Other 98% of the water is going to be lost as transpiration. So we know very well that whatever is the intake of water by that particular plant will also be equal to the rate of transpiration. So yeah, so this is what we are having the method as. Now we have to note here that photometers are of various types designed by various scientists to study and measure the rate of transpiration, namely farmer's photometer and Ganong's photometer, which helps to measure the rate of water intake by the plant. So for intake of the water by the plant, there are two. One is Ganong's photometer, what we are studying now, and the other one is farmer's photometer. So these are two which is going to check the intake of water by the plant. There is Darwin's photometer, helps to demonstrate the suction force created by the transpiration, and Gowray's photometer to demonstrate the unequal transpiration from the two surfaces of dorsiventral leaf. So there are another two kinds of photometer. One is Darwin's photometer. Now Darwin's photometer is going to help you to find out the suction pressure which is generated by the transpiration. So due to transpiration, whatever is the suction pressure which is created is going to be measured by Darwin's photometer. And Gowray's photometer is going to tell you that what is the difference in the transpiration which is going to take place on the upper surface of the leaf and the lower surface of the leaf. Which one will be more? That is going to be understood by the Darwin's Gowray's photometer. So Gowray's photometer to demonstrate unequal transpiration from the two surfaces of dorsiventral leaf. So that is up and down movement of up and down portion of the leaf. Now we understand the Ganong's photometer. So let's start with the Ganong's photometer. A twig or suitable plant cut with the sharp knife is fixed in an apparatus as shown in the figure. So let's understand the figure first. So in the figure we see that it is divided into two sections. One is a beaker. Now this beaker contains colored water. So this is a beaker containing colored water. And this colored water, there is one delivery tube which is connected to this particular place. And then this delivery tube is going to pass like this, completely straight. Like this. And then it is going to end up into a kind of not a funnel exactly, but a kind of an opening which will be having a bigger opening like this, right? And into this particular part will be a cork, and to this cork over here will be fixed a plant. So a plant will be a twig of the plant, or we can say a small twig is going to be connected over here. This whole thing is filled with water. And then there is one particular kind of a connection which is over here, which is called as a reservoir. So there is a reservoir with a tap over here. Okay, there is a reservoir tap which is connected to this one, so that you can I'll tell you what is the reason to have this reservoir with the stopcock. So this is the reservoir. Okay, so this is the reservoir, this is the plant. Okay, now over here will be a scale. There will be a scale which is connected. So this is 0, 1, 2, 3, 4, 5 and so on, right? So there will be a scale which will be connected to this particular part, right? This scale will be sliding. You can put it in any particular part. And then to start with the experiment, we are going to lift this thing up till this tube is comes just above water, and then you put it again. So when you put it up and again, you will find that there will be a drop of air. There will be a air bubble which is going to enter inside, and this air bubble goes and travels and places itself somewhere here. So where it places, we are going to slide it and take this as the 0 point. So air bubble will be at the 0 point at the start of the experiment. So this air bubble has entered inside and reached this particular point. Now what is going to happen? This plant is going to absorb some water and evaporation takes place. So evaporation is going to give you some suction effect, and due to which there is water which is going to be intaken by the plant. And when this happens, the air bubble shifts. The shift in the air bubble is the rate at which the water is intaken by the plant, or also taken as the rate of transpiration. Okay, I'll come again with this whole experiment. So it is colored water put over here. The whole tube is filled with colored water till this point. Over here also there is a colored water. This is sealed completely so that there is no evaporation taking place. A plant is put over here, and over here is a scale. Now to start the experiment, you are just going to take this stick, take it upwards and back it again. When you take it upward and put it again, there will be an air which is going to enter inside. As the air enters inside, there will be air bubble which is going to come. Now to see to it that there is only one bubble entering it. So it should be very fast, it should be very quick, so that the air bubble is not very big, neither the air bubble, there are not 2-3 air bubbles at the same time. So you have to be very careful while introducing the bubble into this particular tube. So that is the main purpose, that the air bubble should be properly placed inside. So the entire apparatus is filled with water so that no air space are present. An air bubble is introduced in the horizontal graduated capillary tube, which is dipped into the beaker containing water. This is done by lifting the bent capillary tube along above the colored water so that the air may be sucked in due to suction pull and is again dipped into the water. So you're just putting it up and putting it down. So what up and down एक बार करने से air bubble अंदर घुस जाएगा. As transpiration proceeds, that is, as the water is lost from the twig, a suction force is set up which pulls the water from the beaker and the bulb in the and the bubble in the air capillary tube moves along. So as the water is going to be sucked in, the transpiration taking place, automatically more the bubble is going to move from here to further inside this the capillary tube. The reading of the capillary tube would be given the volume of the water lost in the given time. The air bubble can be brought back to its original position by releasing some more water from the reservoir into the capillary tube by opening the stopcock. So here what you do is that for resetting the whole experiment, you can just open the tap stopcock. तो इसीलिए रिजर्व से पानी अंदर जाएगा तो यहाँ तो जा नहीं सकता है, इसलिए इधर पुश करेगा तो ये एयर बबल को बाहर निकाल सकता है. Or you can even move it to zero. So suppose it happens that you put it over here and the air bubble starts at this point. Then you can just open the side and let it move over here and it come to zero. That's also possible. So that's why the reservoir over here, the stopcock and the reserve, this is the stopcock, this is just the stopper. So this stopcock and the reservoir is used to reset the experiment or to adjust the bubble position inside the experiment. So it has to be noted here that photometer do not measure the water lost during transpiration. It does not measure the water lost during transpiration, but measures the water uptake by the cut shoot. So it is the uptake what is being measured, which as I told you is almost the same as the transpiration. That's why we are considering as the transpiration. So some of the water is used by the cells to carry out other processes. For example, manufacturing of food photosynthesis. The photometer should be made completely watertight and the twig should be cut obliquely to allow larger surface area of the water intake. Underwater to avoid suction of an air bubble into the twig, which will stop the absorption of water into the xylem. So here some precautions need to be taken. One precaution is that first of all it should be completely airtight, because otherwise क्या होगा, यहाँ से vapors भाग जाएंगे, तो you don't want that thing to happen. So it should be completely this whole thing should be completely airtight, so that one air bubble नहीं अंदर air घुस सकता है. There is a possibility of air entering inside, so it should be completely airtight. The twig over here which is taken should be cut obliquely. It should be cut in a slant manner. It should be oblique, like it should be like this. It should be like this. Okay. Now this would increase the surface area of absorption of water from the stem. So वो करने के लिए you need cut it there. Plus it will also, because if it is smaller, there is a possibility of the air bubble getting into this thing and it will not allow the water to enter inside, because air bubble will block the xylem, which is very very small. So इसीलिए obliquely cut किया रहेगा तो surface area बढ़ेगा तो air bubble रुकेगा नहीं उधर. So limitations of the use of photometer. First is introducing the air bubble is not very easy. Yes, limitation is that it's difficult to introduce the air bubble over here. It is not that easy कि हाँ फटा करके किया और मिल गया, ऐसा नहीं है. It is going to take some time, so it is difficult to do that thing. Secondly, the twig may not remain fully alive for a long time. So this experiment has to be done very fast. Otherwise, what will happen? The twig itself, because it doesn't have any roots, so that's why it may die itself. So the twig may die and the experiment will fail. And thirdly, any chance in the outside temperature changes in the outside air temperature may affect the position of the air bubble in the capillary tube. See, in case ये particular area में suppose we are starting at 10 o'clock in the morning and we are trying to keep, and we have too much of a sun entering into that room, and by 12 o'clock it becomes too hot, then what will happen? There is a chances that the air bubble itself will lose its position due to the heat which is generated inside. So heat के कारण the pressure change होगा, pressure change के कारण यहाँ पे air bubble का कुछ भी displacement आएगा. So the position of the air bubble could be varying, or there could be a change, or could be affected due to the outside temperature being different. So that was the different experimental ways by which transpiration is being measured, or it is being seen, or it is being proved. So that was about the transpiration experiments. Now we are going to talk about the kinds of transpiration, and for that we take the next video.